Hi everybody, it's Madison again. Today we are going to talk about epistasis. What is epistasis exactly? Epistasis is when the expression of alleles at one locus mask or modify the expression of alleles at another locus. This is how we can have multiple genotypes that can result in the same phenotype. Here are the 6 different types of epistasis and their ratios. The type of epistasis I will talk about specifically is dominant and recessive epistasis. Dominant and recessive epistasis is a type of epistasis that occurs when dominant at one locus and homozygous recessive at another locus result in the same phenotype. This means that any offspring with a dominant A allele will result in the same phenotype as little a, little b, and it has a 13:3 ratio. Here is an epistatic pathway for the color trait of the lady slipper flowers. There are a wide range of colors that bloom on the unique lady slipper flowers, such as white, yellow, pink, and purple. But since my favorite color is purple, we will look at that as our end product and colorless or white as our precursor. For dominant and recessive epistasis, any genotypes with the A inhibitor or the dominant A allele will result in the same phenotypic expression no matter what occurs at the B locus. If the A allele is homozygous recessive, it can still bloom as the colorless white precursor if the B locus is also homozygous recessive. If the offspring has homozygous recessive A alleles and at least one dominant B allele, it will bloom purple and result in the end product that we see here. We now know that A is epistatic to B in dominant and recessive epistasis. Here is our example of a dihybrid cross. This is what we get when you cross the F1 generation with itself, and it results in a 9:3 to 3:1 ratio. This is what we use to compare our types of epistasis to find the ratios for each specific type of epistasis. Here is my dihybrid cross for dominant and recessive epistasis. Because of our epistatic pathway, we know that any offspring with a dominant A allele is going to be white or colorless. That leaves us 4 other genotypes to analyze. From the pathway, we also know that even if an offspring does not have a dominant A inhibitor, it can still bloom white or colorless if it is homozygous recessive at the A locus and the B locus. That adds 1 more to the white phenotype. This leaves 3 genotypes that will result in the lady slipper to bloom purple, each being homozygous recessive at the A locus with at least 1 dominant B allele. The resulting ratio is 13:3, which proves that this relationship is dominant and recessive epistasis. Now let's look at another test cross. Here we are crossing big A, little b with little a, big B, little b. Once again, we know that any lady slippers with a dominant A allele are going to be colorless or white, same with any that are homozygous recessive at both the A and B loci. That leaves us 4 more that bloom purple. This gives us a ratio of 12 white colorless lady slipper flowers and 4 purple lady slipper flowers, which can be simplified into a ratio of 3:1 for white to purple. Because this is just a normal test cross and not a dihybrid cross, we do not get the specified 13:3 ratio. Hopefully you learned something new about epistasis. Thanks for watching and see you next time.